Welcome to my channel Adhyan with Flow. Let's start from biological nitrogen fixation. In our previous video, we already discussed about nitrogen cycle. Yes guys, we already done this. And now it's time to discuss about how biologically nitrogen become fixed. I repeat, how biologically, with the help of microbes, some prokaryotes, how nitrogen fixation is fixed. Nitrogen become fixed. How? By biological factor. I'm writing here a flow chart, guys. I'm writing here a flow chart. So whatever written here and here, I summarize in a flow chart. I summarize in a flow chart. So here, biological nitrogen fixation divided into two parts. Yes guys, some bacteria, they perform biologically nitrogen fixation, and some cyanobacteria, these are also bacteria. Then ma'am, what is the difference between bacteria and they make their own food, guys? BGA, blue green algae, they make their own food. But in the case of normal bacteria, they can't make their own food. But yes, they make their own food as well as they fix nitrogen also. So I separate this class. I separate this class. Now, bacteria is two type: free living, and symbiotic. They provide nitrogen fixation free of cost, but they have a condition. They said to the plant, give me shelter, after then I'll make for you nitrogen. What they said to the plant, they said that give me your home, give me some place, then I fix nitrogen for you. And they are free, guys. Take it, free of cost. They produce, they make, yeah, take it, nitrogen. But they want shelter first, and after then they provide nitrogen to plant. Now what kind of these bacteria? Free living bacteria, they give, they don't need any plant and anything, they give nitrogen free of cost. ले जाओ भाई ले जाओ. First is azobacter. Yes guys, free living bacteria. Second is Beiji, look it here, Beijeringia. Okay, they have another special feature, they are aerobic, guys. Now next is Rhodopsilum. Okay, Rhodospirillum. They are anaerobic. They are anaerobic. And more example like Clostridium. They are not written here. They also does not use oxygen. Also one of the example like Bacillus. Also they don't use oxygen. So these are the some example of what? Free living bacteria. And what is the example of symbiotic? And they are bacteria. Rhizobium. Yes, you know na, most popular Rhizobium. Where they located? Legume. What is the meaning of legume? Bean plants, guys. Bean plants like pea, soya bean. Remember some cereals, guys, they found on their roots. Second is most important according to competitive exam rank. Yeah, these symbiotes, where they located? They located non-legume plant. Some examples like ginkgo. I think this question asked already. Casuarina and Alnus dominant. This is the dominant tree, guys. So these are the example of bacteria already written here, guys. I give you extra. Okay. Some cyanobacteria like nostoc, anabina. They also free living and they are also symbiotes. Also symbiotes and they are also free living. And third is oscillatoria. This is the free living. This is the free living. Nostoc and anabina, they both free living and symbiotes. Okay, so this is the microbes, their contribution in biological nitrogen fixation. I am giving you extra, guys. I already give you extra. I clear all the terms, guys. I clear all the terms. Okay, if someone asks that what kind of bacteria who contribute symbiotes and free living also, then you have to remember this. This is the very short, guys. This is the master slide for you, and I don't know how much you are aware for this. Okay, look at here. Cyanobacteria such as cyanobacteria, anabina, nostoc are also free living, and nitrogen fixation are also, guys. this used are also means they also symbiotes. Now, symbiotic biological fixation, I already covered this thing. I already written everything here. Don't worry, guys. Okay, now your theoretical question. How nodule form? Explain with the diagram. This is your theoretical exams point of view question. Okay, I'm not reading this whole paragraph, guys. No, I'm not reading the whole paragraph. I'll make you chart for you. Step by step, you can feel how they perform. I'm not reading this chart. I'm making a flow chart for you. And whatever I written in the flow chart, already they mentioned here. Already mentioned. Because when I'm reading this paragraph, maximum people go sleep. Hibernating mode. So I'll make you chart for this. Okay, let's start. Let's start. First, you have to know that what is nodule. Some node-like structure. Look at here. Look at here, guys. This is the plant, guys. Aerial part of plant. I know this is not a beautiful plant. Yes, guys, there is a root. I told you now some symbiotes, they attach to the plant and take shelter in the plant. Yes, guys, they make nodule in the roots of plant and microbes stay here. Why plant allow this thing? Because these microbes provide them what? Nitrogen, guys. These bacteria, inside the bacteria, they provide nitrogen. So plant allow them. Now let's start. Now let's start their mechanism. How this nodule form in roots. Let's start, guys. Okay. First of all, root hairs gives signal to rhizobia. Means, firstly, root hairs, where they located? At a part of roots, they give signal to rhizobia. How they give a signal? They secrete some flavonoids. Secrete flavonoids. So due to signal of flavonoids, what happened? What happened? Then rhizobia attract towards roots. I'll make it for the diagram. This is the assume this is the root here. Root here. Okay. They secrete what? They secrete some flavonoids. And here are rhizobia, symbiotic bacteria. They smell flavonoids. They smell flavonoids and they attract towards the root here. Now they reach towards the root here. Now what happened next? What happened next? After reaching, I'm writing here, rhizobia moves towards the root here. After reaching, they curl the root here. Now the root here become curly in nature. Like this is the root here. And now this become curl. Look at, guys, this is curl. They form hook like structure. They curl. Now what happen next? Now what happen next? Now what bacteria will do? Now what bacteria will do? They enter in this curly root here. And what they make? They form infection thread from Golgi secretory vesicles of root cells. Now after reaching here, they found a cell organelle. We know now cell have nucleus and have cell organelles. After reaching towards the cell organelles, they meet with Golgi body. They meet with Golgi body and they arise some infection thread. And what they do? They infection thread circulate all over the body. Look at here. Here as infection thread is formed and they circulate all over the cell. Now I'm writing here infection thread end of the cell and fuse with other cell. Now clear? Now clear? They fuse with other cell. Now this is clear. Okay, now next. rhizobia release in apoplastic and penetrate the cell's plasma membrane. What they do now? They enter in a symplastic way. Look at here. I told you before. What is the symplastic pathway? They reach intracellular manner. They reach intracellular manner. It does not cross the cell to cell. No, they only reach. Again, I'm drawing for you. They only flow. The infection thread only flow between the intracellular space. Intracellular space. They do not cross. Their infection thread do not cross the cells. Now what happened next? So they penetrate all the cells plasma membrane. And now what? Infection thread, this thread extend, make a branch, make a vesicles. And what they do? Now they form tumor like structure. So this is the way, guys. This is the way how they form nodules. Look at here. Firstly, there are soil particles and there is lots of bacteria. Root hair release some flavonoid, so bacteria can sense this and reach toward the root hairs. They make a curl of root hair. After curling of root hair, they enter in root hair, and what they do, they make infection thread, and these infection thread circulate all over the body plant in the roots. And what they do, they make a nodule, they make a tumor. So this is the whole mechanism of what? This is the whole mechanism of root nodule formation, guys. This is the whole mechanism of root nodule formation, guys. I already make a chart for you, and this is your paragraph. You can relax and easily you can read this. Many plants, I give you example, not only soybean, guys, not only soybeans, clovers, soybeans, alfalfa leaf. Yeah, guys, this is important for nitrogen fixation. Recently this question asked in UPSC, guys. UPSC. Yes. What kind of plants useful in nitrogen fixation? There is one option is alfalfa, lupine. Guys, I'm gonna prepare you for competitive exam. Relax. So there are the example of which kind of plant do this phenomena. Alfalfa, lupines, they are. This question right now, this 2022 5th June UPSC paper, there is a question. What kind of plant to perform nitrogen fixation? Yes, this is the option. So guys, be in the form. I'm gonna prepare you for competitive exam, UPSC level. Okay, so that's all for now. In next part, we are covered about nitrogen assimilation. Okay, guys, we're gonna do nitrogen assimilation, and then chapter will complete. So that's all for now. If you still not subscribe my channel, then please subscribe now, guys. What are you waiting for now? Still you are waiting for any kind of promotions? Subscribe, guys. Subscribe. I'm giving you lots of knowledge. Don't worry. This is the starting. So thank you. Thanks a lot for hearing me. Thank you so much. Keep smiling and revise. If you do not revise, you forget this. Thank you.